I would like to introduce our studies about the mechanisms underlying cardiac lipotoxicity and treatment potential to attenuate heart failure in metabolic disorders. Heart failure is one of the most devastating diseases and remains a principal cause of morbidity and mortality. The prevalence of heart failure is increased in metabolic disorders. About half of people who develop heart failure die within 5 years of diagnosis, despite of the developing treatment in clinics. Myocardial lipotoxicity is featured with intramyocardial lipid accumulation, mitochondrial dysfunction, and oxidative stress, contributing to heart failure and cardiac dysfunction. However, the molecular mechanisms underlying the impairment of lipid homeostasis and energy adaption in the myocardium upon metabolic stress are largely unclear. Our research on cardiac lipids began with an unexpected discovery. To decode the mechanisms underlying diabetic heart failure, RNA-seq was performed on human heart tissue, which detected an increased PAC3, which is a kinase intracellular, validated by Western blot and immunofluorescent staining. To decide whether PAC3 increase is a causative factor inducing the onset and development of heart failure in metabolic disorders, PAC3 was overexpressed in cardiomyocytes in mice, followed by high-fat diet feeding. As a result, PAC3 cardiac overexpression accelerates cardiac dysfunction. Transmission electron microscopy shows more lipid droplets in the heart, accompanied with more oxidative stress. In vitro model of rat cardiomyocytes confirms that activated PAC3 increases lipid accumulation, while inactivation of PAC3 blocks fatty acid-induced lipid droplets. Since activated PAC3 triggers mTOR as 6K1 pathway, we further investigate the mechanisms whereby PAC3 induces lipid overload in cardiomyocytes. Previous studies have demonstrated that mTOR pathway controls maturation of SREBP1C processing via activation of S6K1. So SREBP1C is a master transcription factor governing the expression or upregulation of lipogenesis-associated genes. As hypothesized, activated PAC3 enhances SREBP1C in cardiomyocytes nuclear fractions. Thereafter, the transcripts of lipid-related genes are increased in PAC3 overexpressing cells. However, betulin, which is a small molecule inhibiting the cleavage and activation of SREBP1C, inhibits the gene upregulation in PAC3 overexpressing cardiomyocytes, indicating PAC3 triggers lipid accumulation is at least partially via SREBP1C activation. So to further gain functional evidence, betulin is applied on mice with PAC3 cardiac overexpression. The group with treatment displays improved cardiac function under metabolic stress induced by high-fat diet feeding, with lower lipid accumulation in the myocardium. So previous studies demonstrated that once the ER-localized SREBP1C is transported to the Golgi, it undergoes proteolysis to become an active transcription factor format, entering the nucleus to upregulate the target genes. Thus, balanced dynamic of SREBP1C trafficking is essential to maintain lipid homeostasis intracellular, probably including cardiomyocytes. Based on cell biology knowledge, we know COP1 complex mediates retrograde vesicle traffic in the secretory pathways, specifically in the Golgi or from the Golgi to the ER. Inhibition of COP1-mediated trafficking of SREBP1C may make it to be constantly activated. And COP-A is one of the key components in COP1 complex, whose loss impairs Golgi to ER trafficking of the proteins, and probably including SREBP1C. So to determine whether COP-A plays an important role in the proper trafficking of SREBP1C, it is knocked down in mice using AAV9 system, following diabetic stress induced by high-fat diet feeding and STZ injection to damage pancreatic cells. Both cardiac diastolic and systolic dysfunction occur in shorter term of diabetic stress, consistent with lipid overload in the myocardium detected by TEM. Untargeted lipidomics further show more toxic lipids due to COP-A knockdown, such as ceramides, diglycerides, and triglycerides. As expected, nuclear fractions of the heart tissue contains more active format of SREBP1C in COP-A knockdown diabetic hearts. More convincingly, COP-A knockdown cardiomyocytes displayed more nuclear localized SREBP1C under stress of fatty acids. As a result, more lipid droplets and more ceramides are found due to COP-A loss. Based on the above mechanistic findings, we propose that targeting regulation of SREBP1C prevents cardiac lipotoxicity because more activated SREBP1C augments lipids, while blockage of SREBP1C or its positive regulators could inhibit lipotoxicity in the myocardium, including COP-A maintenance. When we performed in silico analysis of COP-A promoter region, XBP1S is identified as a positive regulator of COP-A, evidenced by immunoblotting and luciferase reporter assay. Although this investigation about the function of XBP1S COP-A axis is ongoing, we firstly uncovered the function of XBP1S in cardiac lipid regulation. So I will now focus on these findings first. Accumulating evidence implicates that XBP1S, as a well-known transcription factor responsible for the ER function, also plays an important role in lipid homeostasis in liver and adipose tissue. We found in diabetic human hearts XBP1S is dramatically reduced. More convincingly and recently, RNA-seq data from our human sample also identified its reduction in human diabetic failing hearts. Afterwards, we identified a novel target of XBP1S in cardiomyocytes, EDAM2, which is an ER factor regulating protein post-translational modification and transport. By chip assay and luciferase reporter assay, it is confirmed the positive regulation of XBP1S on EDAM2 in cardiomyocytes. As we know, heart failure with preserved ejection fraction is predominant under metabolic stress, while in later stage it may progress to heart failure with reduced ejection fraction. Clinical studies have failed to identify effective treatments for HFpEF, which is due to the limited understanding of the underlying mechanisms. Along with reduced XBP1S in diabetic failing heart, as I just presented, EDAM2 is also reduced, while lipid marker is increased. So a question is then raised: does EDAM2 loss play a role in lipid overload in HFpEF model or HFpEF hearts? So we use the EDAM2. So we use the EDAM2 knockout model to show that EDAM2 loss promotes HIF-PAF and the development of HIF-REF, and the two-hit method induced HIF-PAF model, evidenced by diastolic and systolic dysfunction. Both TEM and oil red O staining demonstrate more lipid accumulation due to EDAM2 deficiency in the heart. Untargeted lipidomics show more diglyceride and triglycerides in EDAM2 loss hearts with severe oxidative stress. Next, we assess whether XBP1S EDAM2 axis is against cardiac lipotoxicity upon HIF-PAF-like stress. So to do so, XBP1 is knocked down with or without EDAM2 overexpression in cardiomyocytes as a treatment. Cardiac function and lipids were examined, and high-fat diet and LNAM induced HIF-PAF model. So we can see the stimulation for 8 weeks damaged diastolic function, but XBP1S loss promotes both diastolic and systolic dysfunction. However, EDAM2 overexpression prevents cardiac dysfunction under the same stress. Intracellular lipid levels exhibit a trend that is consistent with the observed functional changes. Particularly, diglyceride and triglyceride are lower in EDAM2 overexpressing hearts. The above evidence indicates the maintenance of XBP1 as EDAM2 axis has preventive effects. Then we also tested whether EDAM2 restoration rescues diastolic function and reverses lipotoxicity occurred in HIF-PAF-like stress. 4 weeks post-stress, EDAM2 overexpression was administered using AAV9 EDAM2 through vein injection. Cardiac dysfunction and lipid abnormalities are both reversed. In terms of the mechanism underlying EDAM2 regulation of lipid balance, we firstly found an association of endogenous EDAM2 and SEC23A and fatty acid stimulation. We know SEC23A is a key component of COP2 complex, which facilitates the transportation of protein from the ER to Golgi, as well as the translocation of enzymes regulating lipid metabolism to the lipid droplets. On the other hand, ATGL can bind to lipid droplet surface, hydrolyzing triglycerides. Its mutations are involved in kinds of abnormal lipid storage in other organs. Importantly, we also found ATGL levels was reduced in the isolated lipid droplets from EDAM2 knockdown or SEC22A knockdown cardiomyocytes, indicating EDAM2 loss damaged ATGL proper localization and function in cardiomyocytes. Furthermore, we confirm that EDAM2 associated with SEC23A mediates ATGL release from the ER and the proper binding and localization on lipid droplets, preventing lipid droplets formation and enlargement in cardiomyocytes. And furthermore, in human iPS cells-derived cardiomyocytes, we also showed long-term fatty acid stress induces lipid overload or lipid droplets in cardiomyocytes, which are also reversed by either EDAM2 overexpression or XBP1S overexpression. So these results support that XBP1S EDAM2 pathway downregulation causes lipotoxicity in the heart and cardiac dysfunction. as the upstream governor, XBP1S could be a potential target to prevent and treat heart failure in response to metabolic stress. Of note, in line with the other observations, RNA-seq and proteomics analysis performed on human diabetic hearts confirmed the pathways involved in intracellular vesicle transport are altered. Since XBP1S has multiple downstream effectors, including ER chaperones, proteins for trafficking, and proteins for protein quality control. So we assessed the key factors involved in protein trafficking from the ER to Golgi first, and found a decrease in ER Golgi intermediate compartment marker ERGIC53 in diabetic hearts, which is evidenced by the Western blots obtained from various human and mouse diabetic models. And secondly, we also found XBP1S is a potential upstream regulator of ERGIC53 in cardiomyocyte, evidenced by Lucifer's reporter assay. So we know ERGIC53 as a cargo receptor participates in ER Golgi trafficking and secretory pathways, but it is unknown whether ERGIC53 is essential for cardiac lipid homeostasis and function in response to metabolic stress. To decipher its function in the diabetic heart, we established ERGIC53 cardiac-specific knockout model. Under a diabetic stress induced by high-fat diet and STZ injection, both diastolic and systolic function are impaired in shorter time. And untargeted lipidomics further show more ceramides, which is a toxic lipid species in heart, in ERGIC53 knockout diabetic hearts, indicating ERGIC53 loss is prone to ceramide deposition in the myocardium. The latest observation gained from cardiomyocytes confirmed that blockage ER Golgi trafficking using various blockers can trigger ceramides in cardiomyocytes, indicating the importance of protein trafficking system on regulation of lipid homeostasis in cardiomyocytes. Thus, further investigation of the mechanisms is carried on in my group. Finally, to conclude our studies, exploration of gene regulation of lipid homeostasis leads to identification of potential targets to prevent cardiac lipotoxicity in metabolic disorders. We have identified that XBP1S is an essential regulator modulating its downstream effectors, including EDAM2, COP-A, and ERGIC53. Lipid homeostasis is maintained through various downstream molecular bases, which is required to protect against the cardiac lipotoxicity and heart failure and metabolic stress. And so finally, I would like to thank my group members for their hard work, and all internal collaborators and external collaborators to support our projects and studies. Thank you.